Basically, if you look at the last 3 years, there has been a lot of development in the immunotherapy against the tumors. Why it is very exciting and why it's very special, because so far all the cancer treatment, conventional cancer treatment, is based on stopping the rapidly proliferating cells, and it does affect the normal cells as well. So there are a lot of side effects and it's not specific. But when you look at the immunotherapy, it's specific and there has been lots of advancement. So today's lecture is actually I'm going to build up information, all the basics that will help you understand that where we have reached so far and what are the different immunotherapies currently approved by the FDA. So basically the outline of the whole lecture would be, we'll start with the tumor antigens first, and then how the immune response developed against these tumor antigens. And then another important aspect of it is that if there is an immune response, why does still a lot of people have tumors? And then finally, the different immunotherapies currently in use. So just starting off with a small case, and it will emphasize or give you the importance of the tumor antigens. So a 66-year-old black male went to a urologist complaining of frequent urination with only small volumes of urine, creating great urgency during the digital rectal exam, and the urologist felt an enlarged prostate with no distinct nodules or abnormal areas. Now, the different investigations that were ordered are prostate-specific antigen in October 2008. It was 3.9 nanograms per mil, and a year before, it was 3.5 nanograms per mil. So, what's the difference in 1 year? 0.4. We'll see how important that is. And bound to free ratio of prostate-specific antigen is 25.8%, and normally it should be more than 23.4%. Now the questions are, do tissues other than the prostate produce prostate-specific antigen?
Could be.
Could be?
No, because it's specific.
Well, the name itself, tells you that it's prostate-specific antigen, so it won't be produced by any other tissue. Should this man have a biopsy, do you think this man has a cancer?
No.
Yes.
Yes.
Could be.
Could be. So actually I will take your answer correct for both yes and no. And why is that? Because it is a possibility that he has a cancer, and it's a possibility that he doesn't have a cancer. Now, how to confirm it? So the investigations that you see here, prostate-specific antigen, 1 year to another, if the rate of change is more than 0.6 nanograms per mil, he's a candidate for biopsy. Okay? And this particular thing, the rate of change from 1 year to another, more than 0.6 is termed as prostate-specific antigen velocity. Okay?
Again.
So the prostate-specific antigen in 1 year was calculated as 3.5. In the next year, it was measured as 3.9. The change from 1 year to another is 0.4. But if it is more than 0.6 nanograms, this change is termed as prostate-specific antigen velocity. Okay, the rate of change. And if it is more than 0.6, he is a candidate for biopsy. But if it is less than 0.6, then probably not. So in this particular case, what do you see?
No.
Then another thing is, so there are different school of thoughts. One group of doctors, they kind of follow this prostate-specific antigen velocity. The other group follow the free to bound ratio. Okay, so if there is a cancer or a tumor, the free prostate-specific antigen rises very rapidly. It's a lot. So definitely if the denominator is more, it's going to reduce the ratio of the percentage. So if it is less than 10%, again the candidate is for biopsy. So these are the two different methods that you can use the tumor antigen for monitoring or planning your next move. So this is just to emphasize that if you use prostate-specific antigen velocity or you use the bound-to-free ratio, you can decide your next move, whether you are going to do the biopsy or you are not going to do the biopsy. Now normally tumor antigens, they are broadly classified into two groups. Either they are tumor-specific antigens or they are tumor-associated antigens. Now if you think of ultraviolet light or different chemicals that can induce mutation in the genes, they will result in specific type of tumor antigens, and they will be very specific. While on the other hand, I'll give you the example of the oncofetal markers. For example, alpha-fetoprotein, which is used in hepatocellular carcinoma. It is normally produced by the developing fetus, but in hepatocellular carcinoma, the tumor cells start producing it. So it is associated with the tumor, but specifically it can be produced by the fetal cells. And another example is viruses, the oncogenic viruses. They can enter different types of cells. We'll just take two examples, Epstein-Barr virus or human papilloma virus. Both are strong oncogenic viruses. Whichever cells of the body they enter, they are going to produce the similar type of protein, because the virus will encode a special protein, whether they enter the epithelium of the nose or the pharynx or a B cell. So in the end, the antigen will be same. So it's not specific for every tumor. The virus is same, the antigen will be same, whichever body cell they enter. While in ultraviolet lights or chemical mediated antigens, the antigen is going to be specific every time the mutation occurs. So this is just to differentiate between these two broad categories. Now, there are certain other examples. We'll specifically see all of them. So the first one is a normal protein which does not have a response to a T cell. The second one is mutation. Now that mutation could be a point mutation, chromosomal translocation, or a deletion. Any of that will result in a new protein, and I told you that is going to be a tumor-specific antigen. Then, if there is mutation of the proto-oncogenes, they will change into oncogenes, or of the tumor suppressor genes, so the cell cycle is disturbed. It is no more regulated properly, and it will keep on multiplying and will result in different tumor proteins. An example is RAS is a oncogene, while P53 is a tumor suppressor gene. So their products are going to be specific. Then sometimes the proteins are produced by the cells, but in tumorous conditions its quantity is much more. So for example, normally melanocytes of the skin, they are going to produce melanin, but in melanomas the tyrosinase production is very high, and it's basically a chemical that helps in the synthesis of the melanin. So its production is overexpressed, its expression is too much, and that's why it is a tumor-associated antigen. And oncogenic viruses, I already told you, Epstein-Barr virus normally causing infectious mononucleosis, but in many conditions it can infect the B cells and can result in lymphomas. It can infect the epithelium of the nose and pharynx and result in nasopharyngeal carcinomas.
The myeloma, the antigen is the tyrosine.
Yes.
And it's normally found in the body or not?
Yes, its quantity is so less that it doesn't, so there is tolerance against it. It stays in the local environment. But in tumors, its quantity is too much, it's produced in a huge quantity, it is released, and that's why the immune response against it is developed. It's a good question that even the normal products, for example, there are a lot of tumors in which the normal product of the body, when overexpressed, it's going to have an immune response against it.
So should we expect
Even, for example, prostate-specific antigen. Normally, when it is produced, it's in low quantity. But as I said, when the free to bound ratio will change. Why? Because the free quantity of prostate-specific antigen will increase and it will leak out into the systemic circulation.
the myeloma should be expected to the myeloma autoimmune disease against the tyrosine in the whole body or not?
Well, you cannot say, so the response is autoimmune, but actually the root cause is tumor. There is no melanoma, it won't produce that much tyrosinase, and then there won't be a response against it. So the root cause is tumor, that is causing overexpression of the tyrosinase. And then the human papillomavirus is associated with the cancer of the cervix in females. Now that we know all these different categories of the tumor antigens, the next question is it possible to identify tumor antigens?
Yes. Yes.
Completely? Any tumor?
No.
Not all of them, but a lot of them. And that's why all the advancement that we see today, later on I'll give you a lot of examples, a lot of helpful development, and that is based on knowing tumor antigens, knowing the immune response to the tumors, and also the evasion mechanisms. So for example, first thing here what we do is, there's a patient of melanoma. He has a lesion on the skin. You can take out peripheral blood lymphocytes from these patients. Okay, so they are taken out. The blue cells that are seen in the tube, they are mononuclear cells, and also the melanoma cells are resected out and purified as well. So they are separately taken out. Now you might think that all the lymphocytes that we took out from the patient, they have specificity against a lot of antigens. They are not only recognizing the tumor. So what you have to do is co-culture them. So there is the melanoma cells, you put all the group of lymphocytes that you found in his body, put them together with the melanoma cells, and the ones which recognize the tumor, they will bind to the tumor cells. The remaining which don't bind, they don't recognize. So you just wash them off, and then the ones that were bound, you increase their number. So on the right side, with the blue cells that you see is, they are cytotoxic T lymphocyte. So any CD8 cell which is specific against any antigen and it is activated, that is called as cytotoxic T lymphocyte. Otherwise we just call it CD8 cell. So now we have got a cytotoxic T lymphocyte group. In phase 2, what you do is, so normally a lot of groups that are working on tumors, they develop their own cDNA libraries. So for example, whoever have the cDNA libraries, they can have a cDNA template that can be transfected into MHC class 1 cells, normal MHC class 1 cells. Why do you think they did that? What is going to interact with CD8 receptor? MHC class 1. And MHC class 1 is present on all nucleated cells, yes? So if you have a healthy nucleated cell, so you assume that it has a MHC class 1, you take a melanoma gene, a specific melanoma cDNA, and transfect this cell with that. So this cell is going to express that melanoma antigen as well as on the surface of MHC class 1. Okay? So now here we have got 3 examples. First, second, and third. The bottom one is the only one that is recognized by this cytotoxic T lymphocyte that we took from the patient. And whenever the cytotoxic T lymphocyte recognizes antigen, what it is going to do? It's going to kill that cell. That's why it is called as cytotoxic T lymphocyte. So the first 2 are not recognized by this cell, while the last one is recognized. So you already know the gene encoding that tumor antigen, you know its sequence, and you know that this particular lymphocyte which was taken from the patient, it recognized it, so you know the sequence of the tumor. You get it?
No.
Okay, let's go back. In the first case, there's a patient with melanoma. You took out his CD8 cells and took out his melanoma cells. Okay?
Okay.
Now his CD8 cells are a lot, millions. They can recognize anything, but you want the only ones which recognize the melanoma.
Okay.
You put them together, wash the extra cells, just the specific CD8 cells which recognize this melanoma, you have them.
Okay.
Then you put IL-2 in them, their number increases. Okay. Now you keep them at side, and then there are a lot of scientists who are already working on melanoma, for example. They have their cDNA libraries of the possible sequences. Okay. Now again you take normal healthy cells which have got MHC class 1 on their surface, and each one transfected with a different sequence. Let's say 100 different cells were transfected with 100 possible different sequences of melanoma. Now the army that you have, you allow them to interact with these melanoma antigens. Okay? Out of 100, only one was recognized. That one is the sequence of your patient's melanoma tumor antigen.
Okay.
All the 100 are melanoma.
Yes, melanoma has different sequences possible because it's a tumor-specific antigen. Every time the melanoma occurs, the antigen sequence is going to be different. So the cDNA library has got, thousands of different sequences. So they are just tested randomly against these cytotoxic T cells. Whichever recognized, it's going to cause the lysis of the cell. And then you say, okay, this sequence is of this particular patient's melanoma. Now, what type of immune response should be expected in these patients? Innate cells, natural killer cells, and macrophages will have immune response, and adaptive cells, lymphocytes, and antibodies will have response against the tumors. We'll see them one by one. This is the host, the whole army that works against the tumors. You can see macrophages, helper T cells, cytotoxic T cells, antibodies, and natural killer cell. We'll take all of them one by one. The natural killer cell. First thing is, natural killer cell work by a fine balance between the activating receptors and the inhibitory receptors. Okay, so in the first panel, you can see that the activating and the inhibitory receptors, they are balanced. When they are balanced, the natural killer cell won't do anything. But one special thing which the tumors try is that they sometimes switch off the MHC class 1 expression. That's their way of hiding. So when they switch off the MHC class 1 receptor, CD8 cells cannot recognize it, but natural killer cell will, because that's the alternate arrangement from nature. So these tumors which switch off the genes of the MHC class 1, they will be recognized by the natural killer cell. But there are also other ways in which the activating receptors will be increased. Number one, CD4 cells can produce certain cytokines like interferon gamma and TNF alpha. They will activate the natural killer cell. The IL-2 is very important in activating natural killer cells. So if they are produced by CD4, natural killer cell will attack. If MHC class 1 receptor is lost, natural killer cell will attack. And then another mechanism is, if you remember, antibody dependent. So the tumor cell is bound by the antibody, and then the natural killer cell will recognize it with Fc receptors. That is called as antibody dependent cell mediated cytotoxicity. Okay, so these are the three different mechanisms, either with the help of CD4 or imbalance of MHC class 1, it is downregulated or it's lost, and the third one is antibody dependent cell mediated cytotoxicity. Now for macrophages, normally we have two groups of macrophages, M1 and M2, and they have contrasting functions. M1 are the classical macrophages, they are anti-tumor. M2 are actually supportive of tumor, and they are the non-conventional macrophages. M1 produce the cytokines that will kill the tumor. Number one is tumor necrosis factor. Actually, if you focus at the name of this cytokine, tumor necrosis factor. So it causes necrosis of the tumor. That was its function that was first found out. So macrophages produces tumor necrosis factor, and how it does it? Normally, what the tumor cells, when they are rapidly proliferating, what is their first need? What do they need? Nutrition. And that nutrition is through blood vessels. That's why there is a lot of angiogenesis in the tumors. This tumor necrosis factor causes the necrosis of the blood supply to the tumors. So when the blood supply is lost, these tumors are reduced in size. Other than that, they can also recognize the tumor directly. When they recognize the tumor, either with the help of the antibodies, so the two examples that you see here is, in one, macrophage is recognizing the tumor with the help of antibody. In the other, it is producing the lysozymes, the reactive oxygen species, and the nitric oxide. They all are going to kill the tumor cell directly. So these are the two mechanisms. The macrophages are going to produce tumor necrosis factor, which is going to reduce the blood supply or the nutritional supply to the tumor. The second is they will bind with the help of the antibodies, antibody-dependent finding, and then after that they are going to release their lysosomal enzymes, ROS and nitric oxide, and will kill the tumor cells. While on the contrary, the alternative macrophages, M2, they produces TGF-beta, which actually suppress the anti-tumor activity. And we'll see about that because that is kind of used by the tumors later on. Beta. Now, one specific cell which is involved in the immune response to tumor is CD8 cell, the cytotoxic T lymphocytes. Now, normally you all know that cytotoxic T lymphocyte is a CD8 receptor containing cell. It is going to recognize MHC class 1. Now, normally all the tumors are affecting the nucleated cells, but they have MHC class 1, but what they don't have is the co-stimulatory signal. And two signals are must for activating the immune cells. So first signal is there, but second signal is not there. So how they are going to activate this CD8 cells? For this, you have an alternative mechanism, and that is called as cross-presentation or cross-priming, in which the dendritic cell is going to capture the antigen, present it with the help of MHC class 1, which is present on antigen presenting cell to the CD8 cell. It is also going to present the antigen to CD4 cell, and CD4 cell is going to produce, so I just tell you, at the center is dendritic cells. So it's going to do two things. It's going to activate CD8 cell, it's going to activate CD4 cell. CD4 is then going to produce interferon gamma, and this interferon gamma is going to activate CD8 cell, and this CD8 cell will then multiply and it will attack the tumor cells. This kind of presentation, where the tumor cell itself is not presenting to CD8 cell, but dendritic cell is doing the job for the tumor cell, is called as cross-presentation or cross-priming.
Interferon gamma will be the co-stimulatory signal.
It will activate CD8 cell. The co-stimulatory signal will already be here, because antigen presenting cell has the co-stimulatory signal. It has here, and it has co-stimulatory signal here in the form of CD28, and the counter receptor for it is CD80 and 86. Same as here.
So this is the alternative pathway for recognizing the antigen directly.
Yes. So tumors cannot do it. The antigen presenting cell will do it for them. And as a result, what you have got activated cytotoxic T lymphocyte, which will directly attack the tumor cells. Now there is a small information, like a 55-year-old woman have a breast lump and enlargement of the axillary lymph nodes. Patient was diagnosed with breast cancer. Now why the patient has lymph node enlargement or lymphadenopathy? Migration of tumor cells to the lymph node. Will that cause increased size?
Yes, because the immune response?
Yes.
Because of increased immune response, generation of T cells.
Exactly. So the tumor cell cannot migrate by itself. This figure will explain. So if in this figure you can see, actually the tumor cells are at the area or in the lump. Dendritic cells go and take it, engulf it, and just bring that one antigen of the tumor cell. They will not bring all the tumor cells. And inside the lymph node, when the increased number of cytotoxic T lymphocyte occur, that's going to enlarge the lymph node size. And then these cytotoxic T lymphocytes are going to go back to the tumor and cause the killing of the cells, tumor cells. Now you might be thinking that if we have got natural killer cells, macrophages, antibodies, and cytotoxic T lymphocytes working with the help of CD4 cells, why so many people still have cancers?
Because some receptors are unrecognizable.
So one answer is that some receptors are unrecognizable, very good. Subclones.
Tumor cell may be release an immunosuppression.
Yes, very good. All these things happen. Tumors are very tricky. They know how to survive, and that survival mechanism is called as evasion. Evasion of immune response by tumors. There are intrinsic mechanisms and there are extrinsic mechanisms. We look at both of them. So for first thing is, you can see that the tumor antigen is on the surface with the help of MHC class 1, presenting it to CD8 cell. But first thing is they don't have co-stimulatory signal, so direct activation will not take place. Number 2, as I said, some tumors will down-regulate their MHC class 1 expression. So when their MHC class 1 is lost, again, it will not be presented to CD8 cell. They are also capable of producing glycocalyx, and that can hide the tumor antigen, and this is called as antigen masking. Okay. Other than that, there are production of immunosuppressive proteins. Now, this figure is very important because all these things will help you understand the current immunotherapeutic agents. This CD8 is the co-stimulatory, CD28 is the co-stimulatory signal which is recognized by CD80 and 86. There are exactly inhibitory co-stimulatory signals as well. What are those? CD28, CTLA4 and also PD1. So in a normal immune response, when a pathogen is recognized by the immune cells, it is killed, everything is done, then these two co-stimulatory receptors kicks in and they will stop the immune response. Otherwise, if immune response continue, there is going to be damage to the normal cells. So this one is the inhibitory signal, CTLA4. It is actually cytotoxic T lymphocyte inhibitory 4 receptor, while PD1, this is also an inhibitory signal. Just remember these two because from 2015 to 2017 December, there are major advancement with respect to these two inhibitory receptors. Okay? I'll tell you what are those. No, actually CD28 is present on antigen presenting cell, while the cytotoxic T lymphocyte or CD4 have got CTLA4 and PD1. These are the inhibitory switches. So whenever the body thinks that now enough immune response has occurred, they activate this receptor and this, so no more immune response. These are the inhibitory receptors.
So the tumor activates this inhibitory receptor?
Yes, so tumor normally activate this one and this one. So there is no immune response. Now there are certain extrinsic cellular suppression as well. What are these? First, as I said already, macrophages class 2 alternate. They can produce TGF-beta that also have got an inhibitory effect. Then regulatory T cells. They are supposed to control autoimmune responses in the body. So sometimes tumor trigger these regulatory T cells to perform the inhibition of the immune response. And there are certain myeloid-derived cells as well, like the neutrophils and the eosinophils, that can suppress the immune response as well. Now we'll just straight go to the immunotherapy that so far has been achieved. So these include vaccination with tumor antigens. So now you will understand why I told you about the tumor antigens, how you can take out the tumor antigen and find out the possible sequences and all the different steps one by one. So we'll discuss the vaccination with tumor antigens, augmentation of host immunity to tumors with co-stimulatory and cytokines, blocking inhibitory pathway to promote tumor immunity, and non-specific stimulation of immune system. One by one with the approved examples. So we'll start first with the tumor vaccine. Normally you can, this is called as cell-based vaccine, okay? And this particular vaccine has been approved for advanced bladder cancer, urinary bladder cancer, and prostate cancers. So what they do is they take out the dendritic cell from the patient, and then these dendritic cells are transfected with the sequence or the tumor antigen. And then these dendritic cells are put back. So there are two ways: either you can transfect the tumor antigen sequence, or you can keep the antigen with the tumor cell. That is called as pulsing. So either you pulse it or you transfect the antigen. The purpose is that the antigen sequence go inside the dendritic cell. And then when you will put it back, it is going to go and activate the cytotoxic T lymphocytes in the patient. So this has been approved as a cell-based vaccine in the advanced prostate cancers. Number two, it is also a very strong candidate for oncogenic viruses. For example, Epstein-Barr virus and the human papilloma virus. If you transfect the viral antigen into these dendritic cells and put the memory in the patient in advance, there is a possibility that the patients won't develop. So in Epstein-Barr virus, nasopharyngeal carcinoma, lymphomas, human papilloma virus is the cervix cancer in the female. There is a potential for preventing the cancers. On the other hand, as I said, the tumor cells don't have co-stimulatory signals, CD28. So what if you transfect the tumor cells with the co-stimulatory signals? So you take out the tumor cell from the patient, transfect it with CD28 or IL-2, put them back in the patient. So now they have the co-stimulatory signal to activate. So you have to now remember that all the approved things that have been achieved today, they either strengthen the immune response or find out the weaknesses. So in this particular case, what they have done is, so now if you had asked me this question, that there are a lot of tumors in which we don't know the sequence of the tumor antigen, is there a way to deal with them? So the answer is yes. This plan B, it does not require tumor antigen sequence. So in the tumors where we don't know what is the tumor antigen, you can apply this technique. Because what you are doing is take out the tumor cell from the patient, put the co-stimulatory signals, put it back in the patient. So now it's going to directly activate the cytotoxic T cell. So you don't require the antigen of the tumor. And this also has been approved in renal cell carcinoma. Then IL-2. Sometimes what you can do is take out the lymphocytes, peripheral blood lymphocytes from the patients and put them in the presence of IL-2. What IL-2 can do, they produce a special type of cells. So a natural killer cell, normally, if it is put in the presence of IL-2, it's going to convert them into LAK, lymphokine activated killer cells. They are very strong and very active cells. So if you just do this, or you do systemic injection, give the patient IL-2, lot of IL-2, that's going to do what? It's going to activate their natural killer cell, which will start killing the tumor. So I told you the balance between inhibitor and activator. So you are giving the activatory signal to the natural killer cell so that they can attack the tumors. Now, there are also passive immunotherapies. So in case of melanoma, you can take the lymphocyte from the patient, just put them with IL-2, increase their army, and put them back in the patient. And what they do is, they reduce the size of the tumor, and it's been practiced in melanoma patients. Now, monotherapies are also, it has a history now, that rituximab, which is anti-CD20. Do you know what is anti-CD20? It's a monoclonal antibody against CD20 receptor, which is present on B cells.
B cells, yes.
Yes, and it will kill the B cells. Why? Because whenever antibody binds, it's going to activate the classical complement pathway, complement proteins are produced, MAC formation, and it can destroy the tumor. Or it can activate natural killer cell with the help of antibody, it can activate the macrophages with the help of antibody, and it will be recognized by the macrophages or the natural killer cells. Now, rituximab is FDA approved long ago for non-Hodgkin's lymphoma, especially the low-grade non-Hodgkin's lymphoma. And this graph will actually present to you that how good the survival rate is in the patients of non-Hodgkin's lymphoma. So in the 2 years, there is great achievement. 69% of the patients who receive rituximab, they show survival, even despite of low-grade non-Hodgkin's lymphoma.
Rituximab will increase the antibodies?
It will actually, rituximab is antibody. It's a monoclonal antibody. So it's a engineered antibody. Normally, in previous times, the antibodies were prepared in the mouse, but later on they did genetic engineering and prepared humanized monoclonal antibody, which specifically recognizes only one target. Rituximab recognizes CD20. So you prepare a lot of anti-CD20, inject them in the patient, they go and bind to the CD20 receptor in the patient. As a result, the natural killer cells, the macrophages, and the complement are activated and B cells are destroyed. And non-Hodgkin's lymphoma, there is B cell increased number which will be destroyed by this particular antibody. Okay? Does it make sense?
Yes.
Okay. One more thing. These monoclonal antibodies. Other examples are, have you heard of Herceptin or heard of this?
Breast cancer.
It's an antigen which is found specifically, a tumor marker seen in breast cancers, and this monoclonal antibody specifically recognizes it. This is also FDA approved in breast cancers. Okay, so if I just write down some of the therapies. Because for me, today's take-home message is, what are the different advancements? What are the treatments that have been achieved? If you remember those, you are possibly going to link them with why they are achieved or how they are achieved, and this whole lecture will make sense to you. So, for example, the example that I gave you for the cell-based vaccine, what was that? So the cell, urinary bladder and advanced prostate cancer. So, cell-based vaccine, then passive adoptive transfer, when you put back the cells, activate them and put them back in which condition it is adoptive transfer. No, the patient was standing in the figure from his melanoma. Then CD20 is against low-grade non-Hodgkin's lymphoma. Then this Herceptin breast cancer. Then there is one non-specific stimulation. I showed you in this slide. You see this D, non-specific stimulation of immune system. Have you all heard of BCG vaccine? Tuberculosis. Bacillus Bacillus palmatourine. That vaccine, if intravesicular in the bladder, if it is injected, it's very helpful for advanced bladder cancers. It has non-specific stimulation of the immune system against the tumor cells. So, BCG vaccine for bladder cancer. Then systemic interferon interferon alpha. If you give systemic interferon alpha, that also has got activation. It has got antiviral effect and it has got anti-tumor effect. The two cancers that are treated with it is renal cell carcinoma and carcinoid tumors. So far, these are the different FDA-approved therapies. But on this side, now I'm going to write just the advancement in the last 3 years, 2015 June till December 2017.
How does receptor?
It's going to target these tumor markers in breast cancer. So I told you that tumor cells, they can activate PD-1 and CTLA-4. These are the inhibitory signals. So they will be activated by the tumor and immune response will be stopped. So scientists started to think that what if we block these receptors? If we block it, the tumor cells won't be able to activate them. So the concept is this. Normally tumor cell uses it, actually abuses them. They use them to stop the immune response. So why don't block them with antibodies that are able to block these receptors? So once you block them, the inhibition will not take place. So that's why they named them as checkpoint inhibitors. So today, these are called as checkpoint inhibitors. PD-1 is used now in metastatic melanoma. So now PD-1 as a checkpoint inhibitor, it's been used in metastatic melanoma, any non-resectable solid tumor. Non-resectable. So when the surgeon look at it, they say the metastasis has occurred so much, we cannot take it out. So in such tumors, it is helpful. Non-squamous cell carcinoma of the skin, it has also been approved in that, and colorectal carcinoma. And this is minded, it's mainly in these 2 years, because they were all in clinical trials in the last 10 years, and finally in the last 2 years they have all been approved. While CTLA-4, it's also very helpful in metastatic carcinoma, metastatic melanoma, or advanced stage melanoma. So that is all for today. If you have any questions,